Hello, everybody, wherever you may be. This is the introduction to IVRT webinar presented by Radez and supported and sponsored by Teledyne Hanson. Before we get started, let's take care of some housekeeping items. Although you're on mute, you can submit your questions anytime by using the features at the bottom right side of your screen, the Q&A section. We'll do our very best at the conclusion of this presentation to answer all of your questions. If we run out of time, we'll email you directly. If you're having trouble hearing or if there are any connection problems, we suggest you log off and then log back on. And you should also note that this presentation will be available for replay on the Teledyne Hanson YouTube channel. Okay, let's get started. My name is Keith Hammond, and I'm the Vice President and General Manager of Teledyne Hanson, a premium manufacturer of diffusion and dissolution testing apparatus here in Los Angeles, California, in the United States. In addition to the products and analytical services that we offer, Teledyne Hanson is committed to supporting the global pharmaceutical industry with educational materials such as this one. Please check the Teledyne Hanson website and social media channels often, so you don't miss any of this great content. Since this presentation is an introduction to IVRT, or in vitro release testing, it's important that you have a grasp of the tools and technologies available by Teledyne Hanson for semi-solid formulation development and API release testing. The component at the heart of our diffusion systems is our patented Phoenix dry heat diffusion cell, which we developed really as a great improvement over the traditional Franz cell, which was developed back in the early 80s. With all of our work going back to the 80s, we found, really that RSDs over 15% could vastly be improved with better mixing, a larger diameter sampling arm, and a high precision orifice exposure area. The result of our work produced this Phoenix diffusion cell, which creates a fully homogeneous receptor solution throughout the entire cell, including the sampling arm, especially when compared to traditional or other cell designs that are on the market. With all things being equal, this should drastically improve the ability to produce uniformly discriminating results. The Phoenix diffusion cells can be placed in our small dry heat DB6 manual sampling platform, which can be ran in simple mode or with full security, audit trail, printable reports, etc. For automated sampling, the DB6 manual system can easily be placed inside of our robotic diffusion station, or what we call the RDS, which will automate the sample withdrawal and media replacement process, which requires very few changes to your method since it mimics the manual method. And the Teledyne Phoenix cells don't require any circulating water bath due to the dry heat technology, which is incorporated into both of these platforms. Our featured speakers today are from Radez, which is short for Rational Design of Formulations. Radez is a really great CRO that supports the pharmaceutical and cosmetics industries with consulting, development, and analytical services focusing on semi-solid and liquid dosage forms. Their laboratory operations are located in Hamburg, Germany, but they support customers and products from all over the world. Our speakers are from Radez, Mr. Sasha Gorison, who heads project management and laboratory operations, and CEO Dr. Michael Herbig. Although these guys have decades of experience and have successfully worked through so many challenging projects, today's talk is really intended to cover the very basics of IVRT, since more and more pharma development companies are starting to work on semi-solid and other topical dosage forms. So with that, I'll turn it over to Michael.
Thank you, Keith. My name is Michael Herbig, and I will present some general backgrounds on IVRT testing. Later, my colleague Sasha will then take over for some practical examples and tips. First, and briefly, some information on our company Radez, so that you know the background of our activities. We're a development service provider for the pharmaceutical industry based in Hamburg, Germany. we develop liquid and semi-solid formulations rationally designed. That means based on a systematic understanding of the underlying principles. Our focus areas are the development of formulation prototypes, both for novel molecules and generics. We do process development and scale up. We're capable of developing powerful analytical methods also for challenging actives and excipients. We do prediction and optimization of stability. We provide IVRT testing and also in vitro ex vivo performance testing. And we do troubleshooting for existing products and methods. So what are we going to discuss today? First, we're going to look into what is diffusion. Then the important differences between release testing, the focus on today's presentation, and permeation testing will be discussed. We're going to present important aspects of the IVRT receptor cell media and a phenomenon called back diffusion. We're going to present advice on membrane selections and how the release rate is determined. Then there will be information on the validation of IVRT methods and on how to validate the generate data with regards to the comparison of test and reference formulations. At the end, there will be the opportunity to discuss some questions. So what is diffusion and why does it matter? Diffusion is actually not what is determined in an IVRT testing, but it is the underlying process. It is defined as the process resulting from random motion of molecules by which there is a net flow of matter from a region of high concentration to a region of lower concentration. The permeation of perfume of a flower in a room with still air is an example for that. In the diffusion process, the molecules are self-propelled by thermal energy. So there is no external force acting on the molecules. In this context, it is helpful to also be aware of what is not diffusion. Whenever molecules are moved by other methods, such as, for example, stirring or shaking, it is not diffusion, but a process that is called convection, or more precisely, advection. When a material is transferred from one compartment to another compartment, this is called distribution. This, for example, takes place when a molecule permeates from a cream into the stratum corneum compartment of the skin. So this was about the background. Now, what is actually investigated in an IVRT experiment? First, how it is investigated. Typically, at least for studies that are used for regulatory purposes, diffusion cell testing is conducted according to the USP monograph 1724, Semi-Solids Products Performance Testing. IVRT assesses the rate at which an API is released from a drug formulations matrix. It is used for the investigation of semi-solid dosage forms, such as creams, gels, ointments, emulsions, and suspensions. It's important to note that it is not appropriate for liquid formulations, which represent true solutions, as here, no further release from the matrix takes place. Also, in some gels, diffusion may not be rate limiting. For other gels, however, it may be. This depends on the viscosity and the kind of interactions that can occur between the thickening polymer and the active. IVRT can be, and typically is, a sensitive and discriminating method that is responsive to physicochemical changes in a formulation. For example, the dissolution of particles as a function of their size, shape, or polymorph. Another example is the distribution of actives between phases in a formulation, such as the oil droplets and the water phase, or also the viscosity of a formulation. With regards to viscosity, it is important to understand that semi-solid formulations typically show a non-Newtonian flow. That means viscosity is dependent on the shear rate. In the context of release and permeation, the viscosity at rest, or so-called zero shear viscosity, is relevant. This can be measured with high-performance rheometers and is different from values that are typically generated by the Brookfield viscosimeter type. The release rate is typically a critical quality attribute of a formulation. On the other hand, IVRT is not a surrogate for testing bioavailability. It is not expected to correlate or be predictive with skin bioavailability. Bioavailability is complex, and a typical rate limiting step is rather the permeation of the stratum corneum than the release. Briefly, some mathematics of diffusion. As mentioned, diffusion is the underlying process in release testing, so it should be constant during the experiment. Diffusion in liquids can be calculated by the Stokes-Einstein equation that is shown here. Apart from P and the Boltzmann constant, there are three variables. First, the temperature. Therefore, the temperature needs to be tightly controlled during the experiment, as already mentioned by Keith. Then there is the dynamic viscosity, which is typically formulation dependent. In addition, it also has a dependency on temperature. Therefore, the relationship on temperature is not always linear. Third, the radius of the particle, or typically molecule that diffuses, is important. For a given active, of course, this is also constant, unless there are some small aggregates or associates of molecules that dissolve over time. Now we look at some critical factors of the experiment. In general, IVRT experiment must not restrict the diffusion of the active pharmaceutical ingredients from the donor chamber into the receptor medium, or at least influence it in the least possible way. Filter membranes used should be inert and affect the diffusion process as little as possible. Membranes have only the function of holding the drug formulation physically separated from the receptor medium without blocking the diffusion of the API. As mentioned, temperature should be controlled as tightly as possible. Traditionally, temperature control was done by water-jacketed cells connected to a heated water bath. However, in such a system, there is always at least a little temperature gradient. More advanced systems control the temperature directly through a dry heating block, which can be very precisely controlled. This is done in the Phoenix dry heat cells that have been presented. Stirring must be sufficient to prevent the formation of concentration gradients within the receptor cell and must ensure a rapid homogenization of the entire receptor cell contents. This is a photo of the system we use in our lab. It's the Phoenix RDS station that was already presented in all the features, like, for example, the dry heating plugs and the robotic platform. Now we're looking into some differences of release testing and penetration testing. In fact, this is a frequent question. What are the similarities and differences between release and penetration testing? and permeation testing, to be precise. Similarities are clearly in the setup of the instrumentation system. It may be that the only difference, in fact, is the membrane used. Also, the time points of detection and the analytics may be similar. The purposes, however, are very different. The goal of an IVRT test is to determine the release rate at which the API diffuses from a given formulation, regardless of further context of biological barriers. It's a standalone test. In vitro permeation testing, on the other hand, is focused on the performance of a formulation with regards to its ability to provide bioavailability in certain skin compartments or with regards to systemic exposure. On the next slide, the differences will be discussed in some detail. So for the goal of the test, IVRT is determining API release rates. IVPT measures permeation. It provides information on pharmacokinetics. The purpose of the test is to assess product quality and consistency for IVRT and to assess performance and safety for IVPT. As mentioned, the membrane material is different. Filters on the one hand, biological membranes or tissue on the other hand. In general, the variability of the membrane is higher for the permeation testing, especially if the material comes from different donors. For the donor chamber, IVRT is typically used with an occluded top, and IVPT typically or often is unoccluded, exposed to air. For IVRT, a finite dose is applied, so an excess amount of formulations, so that no transformation of the formulation takes place. For permeation testing, often a semi-finite dose is used. Ideally would be finite, but this is typically difficult for the handling of the very low amounts that would be necessary. Here, typically a transformation of the formulation in a secondary or tertiary formulation takes place. The graphical output is a release rate, slope of a curve for the IVRT and a flux profile for the IVPT. There are also some differences with regard to receptor cell media and the detection range. For the drug product and receptor media interface. With regards to an expected in vitro in vivo correlation, the IVRT test is not designed to directly correlate with in vivo performance. The IVPT, however, is expected to correlate with in vivo behavior. And with this summary of the differences of the two methods, I hand over to my colleague Sasha for the presentation of the practical examples and tips.
Yeah, thank you, Michael. The receptor medium used in an IVRT experiment is most often a non-physiological mixture of a buffer and a co-solvent like ethanol or a polysorbate 80 to ensure sufficient solubility of the API. The solubility in the receiver medium should be 10 times higher than the maximum expected concentration at the end of the experiment to ensure sink condition. Under this condition, the solubility has virtually no effect on the release rate. Due to the non-linear solubilization effect of alcohols, a proper solubility screening is necessary. Keep also in mind that the pH value of the receptor media could have a significant influence on the solubility, and that the addition of co-solvents could shift the pH. Ideally, the pH of the receiver medium and the formulation is equal, and the used buffer strong enough to keep the pH constant until the end of the experiment. This slide demonstrates a practical example from our lab, how to calculate the required solubility. Assuming a typical application of 300 milligram formulation with 0.1% API. This means a total amount of 300 microgram API are applied. With a receiver compartment of approx 20 milliliter, the maximum concentration is 15 microgram per mil, and to fulfill the requirement for sink condition, the solubility should be equal or less than 10% of the solubility of the API in the receiver media. So we need the solubility of 150 micrograms per mil. We tested different samples, and in this case, an isopropanol citric acid buffer mixture in the ratio 40:60 fulfills, in this case with a solubility of 196 microgram per mil, the requirement for sink condition. If different solvent mixtures fulfill the requirement, use the mixture with the lowest co-solvent concentration. Although alcohols are preferred from the analytical point of view, because there's usually no interference, the diffusion of the alcohol from the receiver media back into the formulation is possible. This back diffusion caused a decrease in the receiver alcohol concentration and can reduce the solubility of the API in the receiver medium. This effect is depending on the ratio of applied formulation and the volume of the receiver compartment. The higher the applied amount and the lower the receiver volume, the greater the effect. Different formulation types may be affected also differently by the back diffusion. For example, a suspended API can be dissolved by the infiltrating alcohol, which can significantly increase the release rate and the variability. Due to the higher molecular weight, back diffusion of surfactants like polysorbate 80 is less likely, but the polysorbate could interfere with the API during HPLC analysis. So, of course, we have to properly balance the pros and cons. Membrane selection. After choosing a receiver medium, the selection of a suitable membrane is also important. The membrane itself should only separate the formulation from the receiver medium and should have no effect on the release rate due to absorption or any other rate-limiting effects. Tucker et al. Reported such absorption effects for retinoic acid. The absorption effect could be critical, especially at low API concentrations, and should be investigated during membrane selection. Different types of membranes are commercially available. In our lab, we typically use polyethersulfone, PTFE, or mixed cellulose membranes. In addition to investigate any absorption effects, we run an IVRT experiment with 4 cells per membrane type simultaneously to assess the effect of the membrane on the release rate. In this case, we tested PES, PTFE, and mixed cellulose membranes. And in this case, the highest release rate was observed for PES, which was chosen for the main experiment. But how can we calculate the release rate? The first step is to determine the API concentration in the receiver medium. usually performed by HPLC analysis. The cumulative amount of API per unit area is then plotted against the square root of sampling time. This should be a straight line, and the release rate is the slope of the line. Let's have a look at the next figure to make it more clear. The determined amount released per area is on the y-axis, and the square root of sampling time represents the x-axis. It does not matter if the sampling time is given in minutes or hours, but must be constant during the experiment. The unit hour is probably more common. At least 5 time points, for example, 0.5, 1, 2, 4, and 6 hours, according to the ZUPAC SS guideline, should be taken for analysis. But it depends on the formulation, and from my point of view, more points are recommended, especially for initial experiments. The coefficient of determination, R square, should be equal or greater than 0.9, which is described in the draft guidance on acyclovir as an acceptable value for linearity. At the beginning of my part, I mentioned the importance of the sink condition to avoid any bias of the release rate due to solubility effects. Besides this, the depletion of the formulation, the reduction of the API concentration within the residual formulation over time, is equally important. It is recommended by Teledyne to end the experiment before 30% of the API has been released to the receiver medium. The draft EMA guideline on quality and equivalence of topical products described a release of at least 70% to characterize the release profile. But the release rate should be calculated from the linear portion of the release profile, which is usually within the first 30%. So after development, the method needs to be validated to demonstrate the general suitability. In general, the draft guidance on acyclovir describes the validation process in detail very well. The first validation parameters are linearity and sensitivity. As mentioned before, a coefficient of determination, R square, of greater or equal 0.9 is acceptable to demonstrate linearity. Sensitive means the ability to detect changes in the release rate as a function of API concentration in the formulation. For this, the IVRT will be conducted typically with one formulation at 3 different concentration levels, like 50, 100, and 150% of the nominal API concentration. The observed slopes should be significantly different. This sounds easy, but could be difficult in practice. Imagine the API saturation within the 100% formulation is 90%. If so, it's technically not suitable to prepare an identical formulation with a 50% higher API concentration because it is above the solubility limit. Usually, some slight modifications of the test formulations are necessary to conduct the sensitivity experiment. Besides this, the IVRT method should also be selected. For this, the release rates observed for the 50% and the 150% formulation must demonstrate inequality in comparison to the 100% formulation. In addition to sensitivity and selectivity, the IVRT method should also be specific. Specificity in this context is the ability to monitor the proportionality of changes in the release rate as a function of drug concentration and formulation. For this, the The three release rates from the sensitivity experiment are plotted against the drug concentration, which should yield a straight line, as demonstrated in this slide here. To demonstrate that the IVRT experiment is not affected by small variations in the setup, like temperature and stirring rate, the robustness of both parameters should be tested with 6 cells, usually a temperature variation of ±2 degrees and a speed variation of ±10% of the nominal value is chosen. Afterwards, the 4 additional IVRT release rates should be within ±15% of the release rate determined at standard conditions. This is described in the draft guideline also for acyclovir. And now, data evaluation. How can we compare the results between 2 experiments? When are 2 formulations comparable? According to the USP 1724, we use the IVRT comparison test, also named as Mann-Whitney U-test. This is a 2-stage procedure, but the second stage will be used only if the test fails at the first stage. The IVRT comparison test compares the observed release rates of 2 formulations, the test and the reference formulation. The test passed, which means the test and the reference formulation are comparable, if the 90% confidence interval falls within the range of 75 and 133.33%. Yeah, this sounds difficult to calculate, but is straightforward and easily conducted. There is a good step-by-step explanation also in the ZUPAC SS guideline, but I will describe it in detail in the next slides. To compare a test and a reference formulation, in example, a modified formulation with a marketed product, an IVRT will be conducted with 6 cells for each formulation. An example is given in the slide. So you see 6 release rates for the test formulation and 6 release rates for the reference formulation. And the next step is to build a 6x6 matrix and to calculate the ratio of the release rates given 36 results. So, for example, the first cells you have divided 3.684 divided by 2.908 results in 1.267 and so on. So the 36 ratios. The 36 results will be sorted from the lowest to the highest calculated ratio. According to the Mann-Whitney U-test with a 6x6 matrix, the 8th value in this order is defined as a lower limit, and the 29th value is defined as the upper limit of the 90% confidence interval. Those values are multiplied with 100%, given a lower limit, in this example, of 113.3% and an upper limit of 126.7%. The selected 8th and 29th value is only for a 6x6 matrix. If the matrix changed, for example, one release rate is cancelled due to air bubbles and the matrix is reduced to 5x6, you have to select the correct value according to the Mann-Whitney U-table, which is for a 5x6 matrix, the 6th and the 25th value. According to the USP guidance, those limits should be within 75 and 133.33%. In our example, those values are within the range and therefore the acceptance criterion is fulfilled. If the first stage failed, the comparison test must be expanded by additional 12 release rates for test and reference formulation. The evaluation is exactly the same as described for the first stage, except building a 18 × 18 matrix, the 6 initial release rates plus the 12 new determined release rates, and using, according to the Mann-Whitney U-test, the 110th value as the lower and the 215th value as upper limit. So now we are at the end of the introduction to IVRT webinar. Thank you for the attention. We hope we could explain the basic principles and the setup of an IVRT experiment. Now I hand over to Keith.
Thank you, Sasha and Michael. Well done. This was a great introduction into IVRT. We do have several questions from the audience that have been submitted, and I'm going to do my very best to get to all of them, but the heavy workload will be on Sasha and Michael. Just a reminder for our audience, you can submit questions using the Q&A button at the bottom of your screen towards the right side. So with that, we will start. Here's the first question for the group from Radez. What if the formulation matrix is partially soluble in the dissolution medium?
Okay, I'm going to take this. Well, there are several possibilities of such a phenomenon. Typically, if the formulation is aqueous-based, the say aqueous components, of course, are going to mix. If there's something in the aqueous-based formulation that may be dissolved, for example, by alcohols, then, as mentioned, in addition to the general principle of keeping the alcohol content as low as possible, a possibility may be to change from alcohols to surfactants, if this is compatible with the analytics. Another case could be if the formulation is unaqueous, lipophilic, and for example, it's propylene glycol or propylene carbonate emulsion in petrolatum, what is sometimes referred to as an oil-in-oil emulsion system, then this, of course, is going to affect the release kinetics, and there may be precipitation of the API by the miscibility of the organic solvent propylene glycol with the water phase. So it's difficult to say, to give a general recommendation without knowing the concrete example, but the most important thing is really to identify which thermodynamic and physicochemical processes occur, and to find a justification on what to do. Maybe it's impossible to achieve a linear release rate, or the linearity will only be within the first 1 or 2 hours. Then it could help to increase the sampling time at shorter time points. Yeah, I hope this answers the question. Maybe it is referring to something I haven't thought of, then we could follow up on this.
Very good, thank you. So the next question has to do with membrane. Question is, how does the nature of the membrane affect drug release? For example, hydrophilicity or lipophilicity of the membrane as a factor affecting drug release of hydrophilitic or lipophilic drug.
Yeah, also here it's difficult to give a general answer. From a theoretical point of view, it would be more likely that a lipophilic API shows some interactions with a lipophilic membrane and vice versa. But this is also medium and pH dependent. So as a general principle, it's always recommendable to investigate 3 different membranes and then based on the data generated to select the most appropriate one. It's not always predictable what happens. As mentioned, the medium, the API plays a role, of course. I mean, for hydrophilic membranes, if there is H-bond interactions, they could, say, interact with hydrophilic APIs. In general, the lower the concentration of the API is, the more likely some effect of the membranes are going to be seen.
Thank you. And we're still getting a lot of questions, so thank you to the audience. This is very, very good, and you can tell it's a popular topic. Here's sort of a general question. Assuming the development of a generic drug, when do I need to do IVRT or IVPT or both?
I think it depends on the stage of the development process. From my point of view, the IVRT experiment is the test of choice during the prototype selection, and then the IVPT will follow by the final prototypes, because IVPT is much more complex and time consuming. So this is the IVRT for the beginning and selection, and IVPT followed by the IVPT experiment.
Great. And here's a question, a very good question from an industry colleague and friend, actually. Here's a question for Sasha or Michael. Are you using 30% cutoff of applied API release, or do you go to greater than 85% API release regardless of the deviation slope from the linearity?
Usually when we run the IVRT experiment, we choose more than 6 sampling time points, so that we see a release profile of more than 70% API release. But to calculate the release rate, we choose the linear part, or the linear, the first part of the curve, the linear slope, and this is usually within the first 30%. So yes, we calculate the release rate from the first 30%, but we also demonstrate a release profile of more than 70%.
Okay. The next question is, what are the conditions to consider for IVRT of nanoemulsions?
Very interesting question. Again, I think it's about understanding the underlying processes. So we briefly discussed the phenomenon of back diffusion. Of course, there is a possibility that water or alcohols from the medium diffuse into the nano emulsion, and potentially that may destroy the structure of the nano emulsion. But I think it's difficult to avoid. The question is then, would this correlate with the situation in clinic or not, where there is some transepithermal water loss? I mean, if it's just about the technical comparison of formulations, then also there, one may consider if the system somehow stays intact as a nanoemulsion, or is transformed or destroyed too much. Another thing to ask is, I mean, in the case that structures from the nanoemulsion would be capable of passing through the filter, then it would not be anymore a release test for the API, but say intact nanostructures would be present in the receiver compartment. And then that would be somehow be against the principle of the test. So I think that also would need to be investigated by some technique that is capable of identifying whether, say, larger parts of surfactants, etc., are intact. Yeah, liquid crystalline structures whatsoever, swollen micelles pass through the membrane. Yeah, it's about really thinking about what happens and does it matter? Does it still fulfill the conditions for a release test of the active from a matrix?
Very good. And we're still getting more questions tonight or today, wherever you may be. And I'll remind the audience, we will certainly answer a few more questions here, but please submit them using the Q&A button that's located at the bottom of your Zoom screen. It's a little bit on the right-hand side. Here's our next question. What is the level of permeation enhancer that is usually studied in IVRT? I think I just asked that one. Next question, why is the square root of time plotted? Sasha, I'm not sure we can hear you. I'll ask the question again. Why is the square root of time plotted?
Can you hear me now?
Yes.
Okay. It depends. The equation to calculate the release rate is linear or proportional to the square root of time. So you have to plot it on the x-axis to get a linear slope. Otherwise, it would be a different line, and it's would not follow the equation.
Yeah, but it's based on the Higuchi equation, actually, yeah, who described this as a general principle of release. I mean, it correlates to the receding boundary of diffusion, and it's a, say, equation based on empirical observations.
Yeah.
Very good. Here's the next question. How do you discriminate? I'm sorry, let me start again. How do you demonstrate discriminatory ability of an IVRT method when 50% versus 100% versus 150% selectivity can't be achieved due to the nature of the product?
Okay.
Okay, back.
Sasha? Okay, I mean, again, it's about providing a justification. I mean, if it's a suspension and it's suspended at 50%, 100%, and 150%, then from a theoretical point of view, you wouldn't expect a difference. Then the concentration may not be the appropriate variable to demonstrate sensitivity. Then an opportunity or possibility would be that it's the particle size, for example. If say 150% cannot be dissolved in a formulation, which is not so rarely the case because typically formulations are saturated to 80 or 90%, then an opportunity would be to increase the solubility in the system by adding some extra solvent, being an oil or an alcohol. It needs to be decided case by case, and a proper justification, I think, needs to be delivered. But in some cases, it's true, theoretically, you would not expect a difference, like, for example, for complete suspensions.
We're going to answer at least a few more questions here. We still have a few more minutes, but for those of you who might have a question and you're running out of time, all the questions that are submitted using the Q&A button, we will do our best to also reply via email if we don't get back to you during this live broadcast. The next question is, it's a long one. Here's the next question. You mentioned that a slope can be removed from the statistical comparison test. So comparing 5 slopes versus 6 slopes, what are acceptable reasons for dropping a cell's slope from the comparison other than nonlinearity of the cell's slope? And the follow-up to that is, can a cell be removed from the comparison by a Q-test proving it's an outlier even if its linearity is acceptable? So maybe there's 2 questions there.
Okay, from the first part of the question, acceptable reasons usually are, for example, you have air bubbles in the cell, or something happened during the experiment. Yeah, this is usually air bubbles, which you see clearly during the analytics of the samples. So you can have a valuable reason to skip this the cell. To remove a cell, even if the linearity is acceptable, and even and there's no issues during the experiment, so I guess this is not acceptable from my point of view. From my point of view.
Thank you. And a reminder for the audience, we are going to answer a few more questions, at least a couple more, I should say. And you will find this rebroadcast on the Teledyne Hanson YouTube channel. Okay, here's the next question. What's the maximum level of percentage of alcohol allowed to use in the receptor solution or receptor media?
From my point of view, there's no clear restrictions regarding the amount of percent alcohol in the receiver medium, but we usually use not more than 50% alcohol in the receiver media. But at the end, it depends very much on the solubility of your API, which and you need, at the end of the day, you need sink conditions. So you have to maybe increase the alcohol concentration more than 50%. But this can also have some effects on the formulation due to back diffusion, etc. So you have to balance.
Okay. This question has to do with ophthalmic dosage forms. Are there any special membranes that might be used for ophthalmic dosage forms for IVRT?
I mean, IVRT is a release test, so the membrane in general should not affect the release of the formulations. So from this point of view, there would not be a requirement to use a special membrane. Of course, if the test should be predictive for bioavailability, I mean, if you want to conduct an IVPT test, then then biological membranes like cornea or reconstituted cornea, etc., would be appropriate here. But for an IVRT test, the principle of membrane selections would be the same one as for semisolids applied to the skin.
Okay, here's a question about an application with the Teledyne Hanson Phoenix DB6 system. The question is, in an infinite dose study on the Phoenix DB6, is it necessary to note down the weight of the formulation applied to the membrane? And is it possible to weigh and apply the dose in a DB6 tester?
It's usually necessary to note the amount of formulation applied to the membrane. At least we do it for each cell, and so the percentage and applies those things. And I'm not sure, if Michael, if I understood the second part of the question correctly.
I think it's a little more of a logistics question about how to apply the dose to the cell tops and then weigh them on the Hanson system. So maybe we'll do a little follow-up with this person directly. But yes, it is possible to do that.
Yes, in general, it's possible. And maybe to answer this, we use positive displacement pipettes, and we test the application, for example, to 300 microliters or so, and test it different times on the balance to see how is the amount, and then we apply the diffusion cells with the exact amount by the positive displacement pipette to know more or less exactly the amount per diffusion cell.
Okay, we'll try and get through two more questions as quickly as we can. Is there a very bad variation? Maybe the question is, if there is a large variation between cells, for example, a 20 to 25% variation for a slow releasing formulation, is that okay? Or does the method need to be revised?
This is a good question. 20 to 25% is quite high between cells. So I would recommend to revise the method and to look for more suitable receiver compartment or something like that to reduce the variation between the cells.
Okay, final question, and then I'll give a wrap up here. Is IVRT an appropriate test for non-aqueous based foam formulations in which the API is fully suspended?
In general, I would say yes. It's in principle, it is an appropriate test, but it's difficult to conduct, to run the test because you have to ensure that the foam is completely collapsed on the membrane and and is homogeneously distributed. And I think the setup of this experiment is quite difficult, but in general, it is. I think it is possible, or it's appropriate.
Well done, gentlemen. Michael and Sasha, thank you so much. And Teledyne Hanson would like to thank, obviously, both of you guys and your organization, RADES, as well as you, all of our viewers and participants, especially those that asked questions. These are really great questions. As I mentioned earlier, there will be a replay of this webinar on the Teledyne Hanson YouTube channel within a few days. You can watch this on demand. For more information on this webinar or to access other Teledyne Hanson content, product information, you can visit our website at teledynehanson.com or find us on any